Okay, so thank you for organizing this this session. So today I'm going to talk about our line of research aiming to develop new intervention essentially for depression and PTSD. We focus on the neurofeedback approach. So I have nothing, no conflict to report, and I acknowledge my funding before I forgot. So here's the outline of the talk which I'd like to share with you. First, I will show you how we actually end up doing the multimodal integration. So I will mention our line of research using fMRI neurofeedback with simultaneously EEG recording, in particular in depression. Then I will talk about our solution for integrating EEG, fMRI data, and other data stream during fMRI experiments. And then lastly, I will show you some proof of concept experiments in multimodal simultaneously fMRI and EEG neurofeedback in healthy and also in depressed individuals. So here it is, our neurofeedback setup. Some of you are very well familiar with neurofeedback is. It's essentially a closed loop with the patients during fMRI experiments. So you can pre-select the region of interest or the whole networks or connectivity or other measures, do some real-time processing and computations, and feed this signal back to the subject in any graphical representation. We are actually using very simple stimulus design, which is essentially a moving bar, which reflects the signal of your region of interest. We focus on the emotional regulation, so we pre-select amygdala. I will talk about our protocol shortly. So, but on top of that, we also integrate and also we spent quite a bit of effort to do actually simultaneously EEG fMRI for quite a while now. So, of course, the advantage of the integration of EEG and fMRI is that essentially it's just you try to merge the strength of two modalities. So it provides you joint high spatial temporal resolution. And also, fMRI is not direct measure of neuronal activity, it has hemodynamic lag, et cetera, lots of noise. But the EEG reflects more direct neuron activity. The EEG neuronal signal is instant, and if you can somehow, also clean EEG signal properly, then you can have a superb way to actually understand spatial temporal dynamics of the brain, as we've shown in many publications from our group. But before I proceed even further, I would like to actually describe briefly our, emotional regulation protocol, which we developed after a few iterations. So we focus on the essentially, it was neuroscience informed. We try to essentially correct abnormal amygdala activity in depression and also in PTSD to emotional stimulus. So essentially, amygdala response is too weak to positive emotional stimulus and essentially too high to negative emotional stimulus. So we were trying to design the protocol which will correct this abnormal hyperactivity of amygdala by essentially trying to teach the patients or subjects to upregulate amygdala, specifically left amygdala, because this explicit emotional regulation theories, we essentially focused on left amygdala, our clinical target. We also have a sham region which has nothing to do with emotional regulation, and it's a left horizontal segment of interparietal sulcus. And also our stimulus design is actually pretty simple. It has several fMRI runs, like rest, practice, neurofeedback runs 1, 2, 3. And then there's some run which is called transfer, where no neurofeedback information is provided. But neurofeedback task by itself, or neurofeedback run, it's a very simple block design. It has a 3 conditions: happy, count, and rest. And during the happy conditions, the subjects see this amygdala signal, which actually reflects current amygdala activity. And subject task is essentially to upregulate amygdala to the target, which is shown as a fixed blue bar. And then we have the subject is asked to recall happy autobiographical memory. And then, we have also another contrast, essentially, and the condition count to make sure that amygdala disengage after this neurofeedback condition. And then we have a rest period on the task, 40 seconds each. And then we repeat this within the runs. But also an interesting feature of our protocol, what we designed, is essentially linear increase in the task difficulty across different neurofeedback runs. So from run 1, we're starting easy, like 0.5% signal changes, and then essentially we increase this difficulty gradually in linear fashion. This actually is a very important feature of this protocol, because actually if the subject really regulates, it's induced some linearity in the data, which allows us to do much better network analysis as we publish and also much better inference about what other brain regions are actually engaged during this neurofeedback procedure. So we run this protocol on many studies, actually. So we started with healthy controls, and we run this in depression and also in PTSD, but I'm actually showing you some summary of depression results. So essentially, it really works. It's enhanced positive emotional experience of the patients, enhanced recall of autobiographical memories, and also, importantly, we're pretty sure that we have a target engagement, that amygdala is really activated, and so people can self-regulate amygdala in proper direction, and also it enhanced amygdala prefrontal connectivity. So we even run clinical trials, and results were very encouraging. So there is a clinical meaningful effect, and also this procedure changed behavior. Human behavior, which is also very, very good, and we are very happy about that. But we also looked, because we did this with the passive EEG recordings, so which led us later on to better integration. We look on the EEG correlates of such amygdala modulation and essentially, enhancement of the amygdala left prefrontal cortex connectivity on the EEG signal. So because we, again, measure EEG simultaneously with this procedure. So we focused initially on essentially asymmetry, EEG asymmetry in the alpha band, right versus left, which was essentially introduced many, many years ago by Richard Davidson. But from EEG experiments only, that essentially if you have more positive frontal EEG symmetry, you have more approach-related responses to traits and, more approach-related emotions and reduced symptoms of depressions. With the notion being, if you have too much, essentially, EEG alpha power of the right side of the brain, then the cortical area, the brain cortex is actually not working as much. So the idea is essentially to enhance the cortical activity on the left, so reduce the alpha power on the left. So, and then, if you have the other way, if you have more negative frontal EEG asymmetry, you have essentially more avoidance-related responses and traits, et cetera, et cetera. So I'm going to show you how the EEG asymmetry looks like during successful amygdala upregulations with the fMRI neurofeedback. So, and indeed, during the happy versus rest condition, you can see that there is actually suppression of the EEG alpha power on the left, which indicates the more activity of cortical areas under those electrodes. So, and indeed, we run all sorts of analysis, including EEG coherence analysis, which shows that there is actually enhancement on the left side during successful amygdala procedure. So here's some example of the essentially raw data. So this is, again, a neurofeedback session. So during the runs, what we would like to see is essentially enhancement shown in red of the frontal EEG and alpha band right versus left again. So anything positive in experimental group indicates that they are correcting abnormal EEG asymmetry, what they actually have in depression. And in the control group, we don't see effect like this. And also, very nicely, we found that people with more depressed individuals, they have more capacity to essentially correct this abnormal EEG asymmetry by essentially doing successful neurofeedback modulation. And also, importantly, you can actually take the signal from the EEG data and then essentially use this infusion fMRI analysis. And then we found the associations and correlations with, say, EEG alpha power signal variation with essentially amygdala and Russell ACC and all sorts of regions involved in motion regulation, and which was very good. So essentially showing the link between EEG and fMRI really for the first time. So how about the EEG fMRI data integration? So that's the main topic of this. So it has, in my opinion and other people's opinions, lots of advantages. So first of all, so what this means, you have to have access to the data stream at the same time from the EEG and fMRI. As you know, fMRI is noisy and EEG is noisy, so you also have to have some, ongoing data noise suppressions of those data streams. So if you do have this, so you can actually, it opens lots of opportunities. Specifically for neurofeedback, we are not limited to showing, for example, the neurofeedback signal from fMRI, but we can show neurofeedback signal at the same time with EEG, for example. And EEG has, this much higher temporal response, so there is no hemodynamic lag, and effect of the neurofeedback on the patient brain is actually almost instantly, right? So this is one of advantage. But you can do all sorts of cross-modal computations in real time, again, if you suppress the noise properly. And I listed other opportunities. We also studying social interaction with hyperscanning. We have 2 fMRI machines fully coupled with EEG. Each machine has an EEG system, so we can essentially cross-feed the signal and access between not only on one scanner, but among 2 scanners. So we're actually studying social interaction that way. So this is our implementation. It looks very engineering, very complicated, so I'm not going to spend too much time on it. But again, the idea is you have to have an ability to essentially capture clean signal from fMRI and EEG at the same somewhere in the software somewhere, and then also incorporate other physiological measures, like in our case, respiratory and bellow, GSR, and all sorts of other measures we can actually capture and have access during experiments in real time. So we run pilot study. I focus here on the EEG and fMRI neurofeedback. So the idea was to essentially see if the patients can control simultaneously amygdala signal, which is shown here on the right. And for the proof of concept study, which we published 4 years ago, sometimes ago, again, we select the beta band, but and this was actually left versus right, because essentially we would like to upregulate the beta also, because the notion is the higher beta is reflecting higher cognitive activity in the brain. So, so we were showing 2 neurofeedback bars, one from fMRI and one from EEG, and the task was both upregulated with the same protocol design, which I mentioned before, recalling happy autobiographical memory. And indeed, the patients can learn to control those 2 signals. We just like we saw this all of the time. On the fMRI side, there was no problem with upregulating amygdala, and also there was no problem with upregulating the EEG alpha power in the beta band. So we also finally finished the study on depression recently, and Dr. Vadim Zotev has the posters with all details about this today, on Thursday, which essentially the idea was that this study was slightly different to design because it was informed by our previous lane of research. First of all, essentially network modulations on the amygdala. So we determine not only target engagement of amygdala, but we also determine circuitry which amygdala interact during the upregulations and neurofeedback action, which essentially involve rostral LCC, left dorsolateral PFC, but we selected for this essentially neurofeedback experiments 2 targets on fMRI side, left amygdala and left rostral ACC. And again, the task was upregulating those 2 regions. But on the EEG side, we selected alpha band, as I mentioned before, shows you the effect of alpha, so that was actually right versus left in the alpha band, and also in beta, left versus right, also to both upregulate. We were just wondering if those patients can, first of all, upregulate, if there's any clinical meaningful effect of such upregulations. So this is the, we finished the study just recently, so it's just, this is experimental detail. For the sham condition, we did slightly different. We used the artificial signal, but I'm getting close to the end, so I briefly sweep through the results, but full details are on Dr. Zotev poster today on Thursday. I highly encourage you to actually talk with him about this. So, the bottom line, the patients can upregulate left amygdala and left rostral ACC. And also we see a really nice upregulations in the EEG signal features which we selected, essentially, which was essentially alpha band and beta. And then we look, of course, looks on the clinical effect and self-reporting on the patients. So we found a significant reduction in state depression, confusion, and total mood disturbance, as well as significant increase in state happiness after this procedure, which was very nice. And then, of course, we look on essentially more depressed individuals like before. More depressed individuals, they have more capacity to upregulate those features. And then, we found significant positive correlation with trait depression, and also we affect the state of depression after procedure. So essentially, more depressed individuals, they have more room for correction with this, which was very nice. And again, importantly, from independent acquired EEG data, we can form regressors for fMRI analysis and then see if indeed, whatever EEG feature you're after has something or anything to do with fMRI responses. And as before, in frontal EEG, in alpha band, we found all sorts of essentially regions and brain regions involved in emotional regulation, which makes perfectly sense. So, I put you some conclusions, but we think, this multimodal combination of the fMRI and EEG has a pretty, I guess, bright future in depression research as a novel interventions and adjunct interventions. And also, ultimately, this approach can provide the way to really design EEG-only neurofeedback system, which is actually verified by fMRI. By doing this fusion and integration analysis. So I stop there here. So if there's any question, I don't know if we have questions now at the end.
You are happy to take the question now, yeah.
Okay.